Yes, our next talk will be by Alexei Nechitsky, who's been a key driver in the field of proteomics by mass spectrometry and has developed very important tools, bioinformatic tools to enable all of the research and all of the work we do every day. So I'm very interested to learn how this can be applied to single-cell proteomics. So yeah, please be welcome.
Well, thank you so much, Nikolai, for inviting me here. It's really great. I'm certainly learning a lot from you, probably way more than what you'll learn from me, but I'll do my best. I'll talk about FracPipe computational platform and how we use it with single-cell proteomics data that we're not generating ourselves. We get it from collaborators, but we like it. We learn a lot from it, and we want to make our pipeline to be the best pipeline for single-cell data. So that's what I'm hoping to achieve here. And I know some people struggle with this. It looks like I'm one of them. Okay. So this is what how FracPipe looks like. If you download it, we just released version 18. It's really easy to use. That's why people like it, because we have this concept of workflows where you can just select the workflow, for example, non-specific HLA, SciLac, TMT, load it, and everything is set for you. And then you load MZML or RAW files. Whoa, this is really sensitive files. And you click run. And that's why it's so convenient. And you can, of course, customize it and save workflows. The new thing that we just released in version 18 is this visualization module that so many people ask us to add, and we work really hard, and now we have it. So we can visualize the protein coverage spectrum, even for glycopeptides, and so on and so forth. So advantages of this platform, before I get into my main part, the single cell, sort of what we learned from single cell analysis, is that FracPipe is super fast because it's based on our flagship search engine called MSFragger. So all applications that require a lot of time, such as non-specific searches, for example, peptidome data, HLA peptidome or endogenous peptidome, it's very, very fast. Of course, it's great for comprehensive PTM discovery through the OpenSearch. I'll talk a little bit about that. And it's interesting for those who are interested in PTMs, but also as a QC quality control, just to see what's happening with your samples. Glycoproteomics, labile modifications, we work a lot with people in the industry who do chemical proteomics as well as in academia, of course. Interactome studies, protein-protein interactions, we have workflows developed for that, including our resource called Reprint EPMS and CREPOM, Saint interactome scoring. Proteogenomics, we optimized FracPipe and MSFragger and IonQuant, all tools for timsTOF data with taking advantage of the ion mobility. Of course, we support all quantitative workflows, and I'll talk about LFQ with match between runs in my presentation today. And for DIA, we build spectral libraries, as well as we have Diane for quantification using those libraries, all distributed, Diane distributed together with FracPipe. So coming to the main part, which is the single cell proteomics, and what I'm learning from this data, again, not generating the data myself, but learning from you and others in the field, is it's really cool data, right? So we have things that we need to think about, such as that it's a really rapid technology development cycle here. So we have to really be very active, and we have to constantly make changes in the software and the algorithms. You know, a year later, our algorithms are going to be totally outdated if we're not working actively on them. And, you know, FAMES, DIA, you know, ion trap or Plex DIA, I mean, you know, it would drive a lot of bioinformatics people crazy, right? But that's the reality of the field, that we have to be really fast here. The sensitivity is an issue, so we need to improve our ID rates, and I'll talk about that. Very interesting discussion. At least I'm always sort of thinking a lot about, you know, how we use boosting samples as part of our quantification strategies, and how we do a rigorous false discovery rate assessment for the match between runs, because they rely on transferring identifications from boosting samples to a single cell runs, as well as just thinking about single cell protocols that are different than other protocols, and there is a variety of different protocols, and we need to better understand this data and see what kind of artifacts, chemical artifacts, PTMs we see in this type of data. And we'll talk about all of this in my presentation. So let me talk about match between runs. I want to say that our pipeline fully supports TMT-based quantification of eye track. I would not have time to talk about that. So I'll focus on label frequency in my presentation. And specifically, I'll start with a label frequency through MS1 with match between runs. And I don't need to explain what it is. It's been mentioned many times, even through these 2 days. And in general, you're all familiar with the concept in that we have a transfer of unifications from what we call a donor run to the acceptor run, where we did not identify that peptide because maybe a mass spectrum was really bad or it was not acquired. So we have 2 publications in MCP that talk about match between runs and LFQ quantification with IonQuant tool. So I'll briefly go through that, focusing on sort of, again, from the single cell FDR perspective, where, of course, this part of match between runs is the key part. So again, what we have is an acceptor run, and we have donor runs. We found peptides that we did not find in this acceptor run, which may be a single cell run, right? And these are maybe other single cell runs, so maybe there are boosting runs from a higher number of samples. And we would like to transfer unification using MOEZ retention time and ion mobility, if it's an ion mobility enabled instrument. And we would like to do it in such a way that we can control the number of false transfers. And this is something that we worked on for several years, and our tool called IonQuant that does match between runs implements false discovery rate control for match between runs. So that's a key advantage of our tool, because I'm not aware of any other tool that does match between runs that actually implements this FDR control. So and how we do that is that we generate sort of decoy transfers. We take a peptide, we take an ion that we identify in the donor run, and then we shift its MOEZ by some value, and actually we try several different values, and then we try to find that ion with MOEZ value that's wrong in the acceptor run, right? And if you find it, and then we score it, and then we know it's sort of a false transfer that should not happen. And if we have a target run, if it's target value, we find that it's called a target sort of match between run and transfer, and then we have a decoy ion, it's sort of a decoy transfer. And we have a statistical model, and then models this and calculates the rates of false transfers. So we still refer to FDR, but it's sort of a false transfer rate rather than false discovery rate. So here is how it's done in a little bit more detail. Well, I should have used this slide because you see it in zoom in here. So a few points here. We actually worked a lot on custom, in creating options for you how to do it in a very flexible way. For example, for each acceptor run, we can allow any donor run to provide an identification, or we can select n closest donor runs that have the highest similarity to the acceptor run, for example. That's one option. Or we can we have some fancy ways to select what donor runs to allow. In most cases, we use all runs, but if it's a really, really large data set, you may want to limit that. And then for each qualified donor run, we align the retention time and ion mobility. And we have a really nice alignment. It's actually the Metamorphosis group developed a really nice strategy for aligning the retention time, which we also implemented for ion mobility, where we use sort of a neighbor-based alignment. So it's not like alignment of the whole run, it's really alignment, so sort of much more local. So it gives a really good alignment. So we really know well what retention time we have to look for this transfer here, because we know the neighbors here and we know what the retention times here. So we kind of really define really well the region where we want to look for that ion in an acceptor run. And also for single cell data especially, we are of course do it. We're not just aligning the single cell runs to each other. We typically generate what we call library runs, which could be a run with maybe 10 cells or 100 cells. I mean, I wouldn't recommend certainly doing bulk cell lysates in this case, but a small number of cells that's more than a single cell. And we can specify in the software that the library run will always be sort of used as a donor run, even if the similarity would not be really high to the acceptor run. Obviously, it will look different in a single cell run. So we can annotate those library runs in the software, and then they will always be used for alignment. And we have a scoring a function implemented for each match between run and transfer of identifications, and we're not using hard cutoffs like you would normally do with some other tools, where you require a certain retention time window, which you don't really know what to specify, or MOZ tolerance. You really have a scoring function that looks at the difference in PPM between the MOZ value of the acceptor of the donor run MOZ value and a signal that we detect in the acceptor run, or we have the retention time difference, again, after alignment, ion mobility, if that's the ion mobility enabled instrument, as well as some other scores here listed on this slide. So all the scores are combined into sort of a single score. And we have the target transfers, and then we have the decoy transfers, and for each we have a score, kind of like what we've done many years ago with peptide profit, and then more recently with DIA umpiring. So we've done this many times, and this is now we're doing for match between run transfer. So the distributions will look something like this, where we have the target transfers, and they have higher scores, but we have also decoy transfers in blue, and then they have lower scores, but we kind of can model our target transfers as a mixture of the two distributions. So the green curve here is sort of the blue one scale to model the sort of the left tail of the target distribution. So through this modeling, we can basically convert the score of the match between run transfer for each ion to a posterior probability that's a correct match, correct transfer. And then using that posterior probability, we can estimate the false discovery rate through that. And we typically apply 1% ion level false discovery rate for transfers by default, although that's something that can be increased, and I will come back to that in a few slides. So we've tested this extensively in many data sets, not only in single cell, of course, but in single cell as well. And this is not a single cell data set. This is timsTOF, whole cell lysate, HeLa cell lysate for replicates on timsTOF from the MaxQuant paper, which is shown on the left with match between runs here. And this is IonQuant. IonQuant is our tool for match between runs and LFQ. And you see that we can quantify more proteins, but importantly, because we control match between runs FDR, in addition to our optimized signal processing, visually, I hope you will see that we have a much better sort of scatter plot. So the accuracy of quantification is better. You have less outliers, just comparing, for example, the green, which is the human, should be 1-to-1 here. And we have, E. coli, which is 4-to-1 in one direction, and yeast is 2-to-1 in the other direction compared to human. So, the green, the human peptides should be around 1-to-1, on this case, proteins. And you have much tighter distribution than with MaxQuant. So this is published in the MCP paper last year. Again, you can look at this comparison in more detail, as well as other comparisons that we've done there. And hopefully you'll see that we have a very good quantification performance, again, because we optimized all the steps in single processing, alignment, FDR control, not only for timsTOF data, but for thermal data as well. So this is single cell data, the same process, and the data comes from Yingzhu lab at PNNL, Analytical Chemistry 2020. So this orbitrap fusion Lumos data for replicates, and 50 cells were used as a boosting run, and we have 4 replicates of single cell. We have 4 replicates of the boosting runs, 50 cells, and we have 11 single cells. Each dot is a single cell, and then we have 3 cells in triplicate and 10 cells in triplicate as well, and blanks, right? Zero cells. So pay attention here to a few things. I mean, of course, as we go from 1 cell to 3 to 10, both MaxQuant and IonQuant identify more proteins, which is obvious. But for MaxQuant, we have without match between run MBR minus and with match between run MBR plus. And the same for IonQuant, except for IonQuant, we have 1% FDR controlled and 5% FDR controlled match between runs. And you can see that match between runs, of course, for both tools gives a huge increase in the number of quantified proteins. We go from 250 to 750 in single cell data on average. but significantly higher, or noticeably higher numbers with ion quant. And again, we know what FDR we sort of have here, where we're not quite sure what we have for max quant. So we have MBR, and match between rounds is obviously a must for LFQ MS1 based strategy, especially for single cells, and we have improved unification, quantification performance compared to existing tools. And this is just one of the tests. So here is another single cell data also used as part of that paper. I'm sorry, this is the second part of the same analysis where basically it's the same data set, but we also show CV across the 11 single cells. And we can discuss this later, but CV is not a perfect measure here, but we have a better CV in ion quant and max quant. Of course, as we apply match between runs, our CV goes up. So that's sort of expected, and it's still less than the corresponding numbers for max quant. But I'll come back to that point later, because this is not trivial how to use CV here, because this is single cells, right? So anyways, this looks good. The second data set gave us a bit more challenge, and we really like that, because this was from Ryan Kelly, and it was Orbitrap Eclipse, but with FAMES, right? And FAMES, as we just talked, it's something that one has to really think about the data and push your signal processing tools, because it's could be a lot of noise, low abundance signal. I mean, you clean up a lot of noise, but you also have a lot of low abundance signal sort of left to deal with, because you lose some sort of signal power in a way. So anyways, what we're showing here is in this data set, it's 3 replicates of single HeLa cell, not replicates again, 3 single HeLa cells, and 3 samples with 100 cells used for library building. So the library here is from 100 cells, 3 runs, and FAMES with 2 voltages. And there are blanks, and I want to draw your attention to blanks here. And if you look at max quant match between runs, so this is without, and this is with match between runs. So we have 557 proteins on average in each single cell measurement, but 79 in the blanks. So, and that goes to 492 with MBR plus, right, in this data. So clearly there is something not right here. When we control, and we have 97 versus 756 without match between runs. But when we do match between runs with 1% FDR for match between run, we go to 153. So, well, it's still noticeable number, but at least we're able to control it compared to max quant, which it just really explodes, right? So we at 5% FDR, this is where we get 480 proteins in blanks, but we get 2,000 proteins in a single cell data. So the point here is that we are really getting we are much more rigorous with what we're transferring from the library runs to single cells and to blanks as based on this analysis. But we're still have comparable numbers even at 1% FDR of what we see in the single cells, even though our error rate is way lower. And that's because we sort of really optimized our signal processing for FAMES data, but also because we have the match between run control, FDR control. So, but this also brings 2 good discussion points that I want to briefly mention. I mean, one is that the false discovery rate estimation for match between run and benchmarking of that is of course difficult, because how do we know what this empirical FDR is, and blank samples are not really necessarily true negative controls, because if I go back here, I mean, clearly they're not totally blank, right? So there is always something in those runs. So we cannot really say, it's not 0. It's not we go from 0 to 10. We already have something even without match between runs. So clearly it's not a real blank. So that's why I think really it highlights the need for statistical control for match between run FDR, like the type of control that we are using. And second one, I also mentioned that sort of quantification CV across single cells, is it a good metric of the accuracy? And we had some discussions earlier, and I think the answer is yes and no. So because cells are different, but still the CV shouldn't be super high. If you start introducing false quantification, and CV goes up, that's a bad sign. But just the fact that we see 25% CV across single cells, that's not necessarily bad because they're different cells. So I think it's imperfect measure of accuracy of quantification that we can use still when we compare different tools. But I'm looking forward to better data sets that can be used for this type of benchmarking, such as those that Nikolai, you mentioned in one of your presentations, I think earlier today. And discussion point 2 is what is the reasonable MBR FDR value to use? I mean, if you do whole cell lysates, you have to be really stringent because you don't really need to, you're already reaching the saturation point in which you can really transfer between different runs because they're similar. You don't need to really, push into the noise there. There is no benefit. But single cell is sparse data naturally, and you may want to really push into high FDR just because you have really so much missing data missingness here. But you only should do that if you understand what you're doing later downstream. So some computational steps that you do downstream can be quite tolerant to error rates and inaccurate quantification, and other methods may not be. So I think it's okay to sometimes increase from 1% FDR in our tools to say 2% FDR, as long as you know how the downstream data analysis can handle the errors. Like if you do pathway analysis, that's probably fine. For example, if you're really interested in specific proteins, you want to be stringent and so on. So now I would like to talk a little bit. How much time do I have? Anyone keeping time? Okay, so deep learning. So we implemented deep learning-based rescoring in a tool called MS Booster that basically takes the output from MS Fragger and then uses predictions of retention time and spectra to calculate additional scores that then go to Percolator. We use Diane for predictions, but we also can use Prozit, and you get huge boosts. And this manuscript now been submitted, if not next week, then soon. We get really huge boosts for HLA peptidome and for DIA data, 20%, much less for bulk digest. So we thought, well, let's see how this would work in single cell data. And we realized that, well, in some data sets, actually, we get some boosts. So this is a single cell data set from this study here, where we got about 10% boost in the number of IDs in single cell, or even a little bit more, 14%, 12% for 5 cells. So 10% boost when we use retention time and intensity predictions after MS Fragger and Rescore. And we looked at the similarity between the predicted spectra and empirical spectra and comparing to the bulk data, and they're quite comparable. So that was good. But then we took another data set, and it's like, wow, that's very different. So we took another data set, that's actually that InJuke data set, and we saw no gain in using rescoring, almost no gain for single cell data. And we were like, what's going on here? And then we plotted the similarity between predicted and experimental spectra, and it was way, way less than what we see for bulk spectrum. So we think that it's because we have, in this data set at least, we have a spectra of lower quality, or they're just different. So our prediction tools actually don't predict the spectra for single cell data from this data set really well. And that's why we did not gain using deep learning rescoring. And in fact, there's a paper from Sam Payne at BYU that actually talks about peptide fragmentation spectra in single cell proteomics, and also suggests that the spectra look different than the bulk data. So we really need to still work on improving spectral predictions for single cell proteomics. So the best spectrum actually, like a really good e-value, but the similarity is not that great to the predicted spectrum. So we have more work to do here. So very briefly, we looked at another benchmark data set and see how FragPipe 18 performs compared to other tools listed here, and we performed pretty well. This is a preprint that published this benchmark single cell data set. But then we also were curious, because we have all these tools for exploring PTMs and artifacts in this data. Like, let's see what our OpenSearch will do using MS-Fragor search engine. That's really, is a perfect tool, and the whole pipeline we have for comprehensive PTM discovery to study this dark matter of proteomics. So we run this tool, and the PTM Shepherd post MS-Fragor processing tool that summarizes all modifications, mass shifts that we find in the data with respect to site localization, spectral similarity, retention time shifts. And this is a manuscript that describes this work published in MCP. So we used MS-Fragor OpenSearch and PTM Shepherd to run that single cell benchmark data set. And we identified that in this data set, there is a modification, I'm not going to say it, plus 40 mass shift. That's very, very abundant, like 6-7%. So that's interesting. And that's, again, specific to this protocol. I haven't seen this particular chemical artifact in any other data sets. So it's specific to Ryan Kelly's sample preparation protocol. And our tools allow us to quickly see that, so that when we went did the search where we specified this particular modification as a variable modification, as well as some other modifications, we improved even more. All I'm saying this just that we have the tools to go into this data and explore what kind of artifacts and things that we see in this data. And it's very useful to understand the technology, the protocols, and often we can actually improve our analysis by doing better searches based on what we learn using this OpenSearch strategy of MS-Fragor and PTM Shepherd. So finally, if I have 1 minute to really wrap it up, a DIA analysis. I mean, this is a big part of what we've been doing for 10 years now. We were the first to propose the direct DIA approach and this DIA-Umpire strategy, when no one believed that you can actually identify peptides directly from complex DIA spectra. So we came up with DIA-Umpire, which is now implemented in Spectronaut, this strategy. Our favorite strategy, and it's all available in FragPipe, is actually to build hybrid strategy. So if you have some DDA data, you could go through regular MS-Fragor, Percolator, and then you have DIA data, which goes to DIA-Umpire, and then MS-Booster and Percolator, and we build a library, and then we use Diane for quantification. Now, more recently, and this is not a manuscript that we're submitting soon, we developed a new strategy called MS-Fragor DIA. That's actually much more sensitive to DIA-Umpire. So it's a different strategy, but it's also a direct DIA approach, and this is sort of our primary workflow now. So and this is some examples of what we get in on low input samples from our Taiwanese collaborators here. This is the manuscript. This is 5 cells. It's not single cell, 5 cells and 10 cells. And MS-Fragor and Diane library free give a very comparable performance, except MS-Fragor DIA is much faster. But note that we use Diane for quantification here. So the identification comes from MS-Fragor DIA, Diane for quantification. We compare it here with Diane for library free and quantification. And I don't know what's wrong with Max DIA. We tried. I'm sure it works better in other people's hands. But this is Spectronaut and DIA-Umpire. So anyways, we have a very fast strategy that gives really good results. And unlike Diane, it does not require predictions of spectrum in advance. So you can run it with PTMs, for example, that you cannot predict spectrum because it does not rely on the predictions. So anyways, we also run it on the single cell data. And this is where we clearly outperform all other tools. And we have a reason for that. And it's because Diane works really well when you have runs that are very, very similar to each other. And in this case, the library is built from, again, 10 cell runs. combined with single cell runs, and Diane just does not do a very good job when we have runs that are very, very different, like 10 cells versus single cell, where in our workflow, it's not a problem. So far in what we see for single cell data, that gives the best results. And it's a good paper also to see discussion about what's the best library for a single cell DIA analysis. So they use 10 cells and bulk samples diluted to 10 cells, analyzed using DIA to build the library, and then go through the single cell DIA data. Instead of using sort of the bulk lysate data that works better with DIA on larger starting amount. So finally, we work very closely with Vadim Demichev, and we have a great relationship and collaboration. So this paper is coming in Nature Communications, where we basically have the workflow that in FracPipe you can do the whole analysis with building the libraries for DIA passive data from DDA passive right now. And then use Diane for quantification using FracPipe build libraries. And this is the bulk HeLa data, but very short gradients. So with Evocept DIA passive flow, we can 5,500 proteins or so in less than 10 minutes. And this strategy has been adopted for single cell. So the Man Lab used this strategy. So they used FracPipe with the library built from DDA passive data from sort of higher input samples using DDA passive, and then Diane was used using FracPipe build library to quantify. And I am out of time here, but I think it's really interesting discussion on how to build libraries for single cell analysis. And I still think building with DDA from a little bit more starting material is ultimately the best sort of strategy for single cell analysis with DIA passive strategy. So FracPipe is a comprehensive next generation computational platform that's available for download at this website. And our team is always open to new collaborations in all areas, including single cell proteomics. That's why in my last acknowledgement slide, you'll see that in addition to my lab, and I want to specifically thank key individuals here, so Franchel Hughes and Felipe Leprovost, and Go Citeo, Danny, Dan, Kevin MS Bookster, and Kylie, a new student, and true, Jeannie and Leo. I mean, I have a really great team. And former members of the team listed here, but I want to really thank our computational collaborators, because we add additional tools to FracPipe, Diane, EasyPQP, Percolator, PPCM Profit, and so on. But also our collaborators who really drive our work. And some of this you recognize here. Inju, Andreas Brunner from the Man Lab really pushed us a lot to develop these tools, and many listed here. Others, and I added Ryan because we already discussed a new collaboration with him yesterday. So I would like to see more of you on this list here. We have to be selective with collaborations, but this is a terrific audience here. So if you have some ideas for collaborations, you're always open to that. So thank you for your attention. If there is time, I'd like to answer questions.
Thank you very much. Are there any questions in the audience?
Thank you for sharing your results with us. I would like to go back to the slide where you compare MaxDIA, FracPipe, and all the other tools on 5 and 10 cells. So it's impressive to see the difference you have here on the previous slide. It's impressive to see the difference you have with Diane and MaxDIA. Do you check the proportion of proteins which were quantified with one peptide in each tool, or?
No, we did not check that, and we typically do not apply like a 2-peptide rule, for example, here. It certainly would not apply probably for No, like with match between runs, for example, we apply typically 2 peptides there. Here we could also apply it, but this data is not filtered to 2 peptides. I don't know what, we had really trouble running MaxDIA in the first place. So, this is not published data yet, and so, you know, probably maybe during the peer review, someone will tell us how to run it the right way. Maybe we will not put it here because something is clearly not right here. Again, I've never used this tool personally. I don't want to say why it's performing like this because other tools are, you know, pretty comparable.
Thank you.
Are there any more questions?
I'll start with a comment and then the question. I think it's great that you're aiming to estimate the false discovery rate in doing match between runs. It's difficult to estimate it reliably. There's more work to be done there, but I commend the effort to estimate it, not just do the matching. And my question is, what is your thinking about peptide-like features in the small receiver samples that are reliably identified but not matched to any identified peptides in the larger samples? Let's say if you have some single-cell runs in which you identify confidently peptide-like features, but they don't have corresponding feature in the donor run.
Okay.
Are there many such examples?
The donor run is the, say, 10 or 100 cell. Yeah. Well, you know, it's interesting. I never looked the other way around. We always look, you know, from donor run to the acceptor run. You're asking a good question. Of course, there would be, certainly suppression effects, you know, the ionization effects, right? I mean, I could easily see how a low abundant signal would disappear if there is another high abundant peptide that's present that would suppress the signal. I mean, you're asking a good question. So yeah, we should see what we find in single cell and we do not find in bulk runs.
Sounds good. Thank you.
Thanks for that. So there's this big development in terms of predicting spectra with DirectDia, or Leonard Martins showed his IronBot workflow. I'm very curious to hear your thoughts about, do we really need to create libraries, or will we get so good at predicting these spectra that we can just predict them and not rely on any libraries?
Well, the way we do it, we do not like creating, well, our strategy is that we believe that we can identify with our algorithms peptide really well without any deep learning, with good mass calibration directly from the data, and then bring a rescoring based on predicted spectra as a second stage. So we don't work with like really empirical libraries that are coming from some other experiments that's completely unrelated. We're relying on predictions. And I think predictions are good, but we do predictions at the last step. So instead of predicting for every peptide before you start the analysis and generating in silico predicted library, we basically do it as the last step. Once for each spectrum, we have a list of highest scoring candidates, then we predict for those. So then that means we can do predictions very fast. That's why we keep our frag pipe fast, because we don't have to wait for an hour to do predictions on the whole data set before doing anything. We only do predictions on what we identify by MSFragger as a top hit, and that's a much smaller number of candidates, right? And we still do predictions. So as far as the sort of building libraries from completely unrelated runs, again, for DDA, we don't do that. For DIA, again, we're building from the data that we're analyzing. So if you're building a library from DDA runs, that means that it's sort of generated as part of the project. We're not typically take DDA data from some completely unrelated runs and try to build a library first. That was kind of the initial thinking about the DIA. So I'm not sure I answered your question correctly, but anyways, that's where my thought went.
Oh, very helpful. Thanks.
Are there any more questions?
Thank you, great talk. Just quick question about your algorithm to predict for the FAMES data with either Eclipse or Explorers. Have you also incorporated it with like trying to predict the CVs, because it make things complicated in algorithm?
Well, you're asking if we are predicting CVs for FAMES data? We are not predicting CVs for FAMES data. When we trace peaks, we certainly trace, fragments of the precursors across the same FAMES being, but we are we're not predicting for timsTOF data. We're predicting the ion mobility, this collision cross section. We are predicting there. However, I must say that we're not gaining much from that, but we're not predicting for FAMES data.
But we can use the FAMES data for that.
You can use the FAMES data, right? So, yes, everything is natively supports FAMES data. Without, you just take the raw file or MZML file, and that's all you need to do. You don't need to split it into FAMES, like file 1 with one collision energy, file 2 with another. You don't need to do that with our tools. But when we do rescoring, for example, we are not predicting the collision, the FAMES value.
Great, thank you.
Thank you very much. Are there any more questions? I think we are, yeah, oh, sorry.
Just a general question. So you advertised in earlier papers that the MS-Tracker algorithm is much faster in the analysis of timsTOF data than, for example, MaxQuant or the PEAKS algorithm. So what is the difference? What is the trick that it is so much faster?
Well, yeah, so yes, it's about 100 times faster than most other tools, and that's because, think about this, when you do this analysis, you take a spectrum and then you take a candidate peptide in a database and you compute the similarity score, take next tryptic peptide in a database, compute the score, the next one, right? So you do a lot of comparisons. We index the database in such a way that take a spectrum, and we basically score the spectrum against all candidate peptides at once. We take the first peak in the spectrum, an experimental spectrum, and see what fragments in the index are matched. Take second peak and see what fragments matched, and increment the score of all peptides that have that fragment, right? So it's like we go through spectrum once and increment the score of all peptides like in one go, right? So we basically, instead of doing the same computation, comparing MOAZ from the spectrum to MOAZ value in the theoretical spectrum many times, we really do it only once. So that's why it's much faster. It's like really basically searching simultaneously spectrum against all candidate peptides in a database. And it's always very clever indexing that sort of we worked for a while, and we really figured an algorithm.